Red? Yeah. Red means on? All right. That's too confusing. That's like the cell phone. I just got a cell phone. And green and red, and they're the opposite of what they're supposed to be, so I'm compromised with a cell phone too. All right. All right. I'm not allowed to do that, or I won't even get through half my talk. All right. All right. So let me start by thanking you guys for inviting me to give you sort of an overview of a system we've worked on for many years, and the title I decided to choose was a playground, ribonucleotide reductases, RNRs, that's what we'll call it, throughout the talk, a playground for controlled radical reactions in biology. Today, I asked the question when I was thinking about a subtitle, how can anyone study? I get asked this question all the time, one system for 40 years, okay? Well, I'm here to tell you that not only me, but all the other R&R co-workers in the field, and there are substantial numbers of these people, I think have made one unexpected and exciting discovery after another in the biology of this system. So it's kept me, and I think the other R&R people, highly amused. And my goal today, since I'm retired, is for 3 or 4 of you at the end of my seminar to come down and tell me that you want to work on the next generation for the next 20 years of R&R, and I guarantee you're going to find something interesting and unexpected. Okay, so there's so much stuff to talk about. I started with 150 slides and I'm down to 74. Okay, and what I'm going to try to do is just give you a few short stories about what I think is interesting about the system after I give you an introduction to the system. Okay, so reductases play a central role in nucleic acid metabolism in all organisms. They catalyze the conversion, in most cases, of nucleoside diphosphates, 4 of them, to deoxynucleoside diphosphates. And subsequent to several rapid reactions, including phosphorylation, all of the monomeric precursors required for DNA replication and DNA repair are supplied by this one enzyme. So this enzyme not only makes these building blocks, but it controls the relative ratios of the deoxynucleotide pools, essential for fidelity of replication and repair, and it also controls the absolute amounts, which helps the cell decide whether it wants to arrest or continue living or send itself into apoptosis, given its vital role in nucleic acid metabolism. And the enzyme, because of the central role, is regulated at every level imaginable. And here's a list of the different regulatory mechanisms. Today I'm going to specifically focus on allostery. So we'll see how that deoxynucleotides can control which of the 4 substrates are reduced. So that's specificity. It can also control activity. It can turn the enzyme on and off. And I would say in the last 6 or 7 years or so, it's clear that nucleotides control quaternary structure of these proteins in ways we didn't really get. And so I'm going to spend some time talking about that today. And in the second half of my talk, since it's on metal-based reactions, I'm going to talk about how the metal cofactors are assembled and repaired, with a major focus of how we think this might happen inside the cell. So you guys out there, you need to use all the modern technologies, CRISPR, Cas, computation, mass spec, to really start understanding nucleic acid nucleotide metabolism inside the cell. We don't get it, so that's up to you. Okay. So the reaction looks pretty simple. It's converting a nucleotide into a deoxynucleotide. We have 4 substrates, ADP, UDP, CDP, and GDP. And what you see from this slide is that there are 3 cysteines in the active site, 1 on the top face, 2 on the bottom face. This is a reduction reaction. And the first irreversible step is cleavage of the carbon hydroxyl bond. And the reduction occurs by the bottom face cysteines, which get converted to a disulfide concomitant with substrate reduction. And so turnover in all organisms requires a reducing a system. Here, thyroidoxin reductase. Some organisms, they have 5 different reducing systems, and you have to figure out which is the correct one, and the rates of the reactions are dramatically different. So you see another cysteine up here, which is a sulfur radical, which we're going to spend a little time talking about. Before studies on RNR, there were no examples of characterized sulfur radicals in biology, and the model is that the sulfur radical, where does it come from, can remove a hydrogen atom, 1', 2', 3' position, to form a nucleotide radical. You go through a long series of complicated, detailed radical chemistry, most of which I'm not going to talk about, and then you get deoxynucleotide, and this hydrogen, for some reason my pointer is not working, the hydrogen abstracted from this position is returned to the same position. So this cysteine, unlike the two on the bottom face, undergoes a transient reduction and reoxidation on every turnover. Okay, so when I started working on this 15 years ago, we knew nothing about structure. First structures came in 1994. But this is our current model, and I'm going to spend the introduction of my talk talking about structure because I think it's front and central now in thinking about RNRs. So there are 3 classes of ribonucleotide reductases, and the classification is based on the Metallo-cofactors. That's why I got invited to give this talk. You might not know that till the second half of my talk, but that was it. Anyhow, they all have 10-stranded alpha-beta barrels. Here's the nucleotide. Here's the 2 cysteines that I showed you got oxidized, concomitant with substrate reduction. They all have a finger loop in the middle of the barrel. At the tip of the finger loop is this cysteine that needs to be oxidized. They also have a glutamate, which you'll see in some of the slides. Anyhow, you have a cloud here. And the cloud, for me, was the most amazing thing about the 3 structures. That regardless of the organisms and the Metallo-cofactors, they all look the same, and the oxidant of converting cysteine to a sulfur radical was hiding under the cloud. Okay, so what are the Metallo-cofactors? Okay, so the class 2 Metallo-cofactor uses adenosyl cobalamin, shown here. In this structure, you can see the cysteine and the cold-2. We don't see the axial ligand. Okay, and the distance here is short, like about 5.5 angstroms, and so the oxidation, we believe, involves hydrogen atom abstraction. Okay, the class 3 enzyme is a member of a radical SAM superfamily of enzymes. The activating enzymes has S-adenosyl methionine and a reduced iron sulfur cluster. Its job is to generate a glycol radical. And this glycol radical, if you pick up the cloud, is sitting right there within 5 angstroms of the cysteine to be oxidized. So again, hydrogen atom abstraction. Today, mostly what I'm going to focus on, I'll have a little bit to say about these, is the class 1 RNRs. These are found in humans and the E. coli system, which has been the paradigm for this for decades. And if you pick up a cloud, what do you see? You see 2 tyrosines. Well, tyrosines are not redox active. So how can this possibly work? What happens to our model? Well, Reichard showed many years ago that there's a second subunit to the class 1 enzymes. And this second subunit contains these Metallo-cofactors. So the class 1 contains a diferric tyrosyl radical cofactor. We'll talk about in the second half of my talk. What's amazing about a tyrosyl radical cofactor, of course, is that it's a really hot oxidant, 0.96 volts. How does the enzyme prevent a cell from killing itself? And one of the functions of the cluster is to make the tyrosyl radical. This radical, if you reduce it with an electron and a proton, your enzyme's dead. So it's key to initiate the radical reaction. I'm going to talk very briefly at the end of my talk about detailed analysis in a rabbit model of the dimanganese tyrosyl radical cofactor we now think is found in all or most pathogenic organisms. And then we have the Penn State 1C system, the manganese 4 iron 3 chlamydia system, no tyrosyl radical, phenylalanine. And so we have different kinds of oxidants that are required to oxidize the sulfur to a sulfur radical, but the structures of these proteins are all pretty much the same. So we need, in this case, two proteins to come together to form that sulfur radical. And very recently, at Penn State as well, they discovered a 1D enzyme. This was just published. They believe it's a manganese 4, manganese 3 system formed by superoxide. And Gavin, Amy told me, has a poster on the 1E, which is even more amazing. Okay, go see his poster. Okay, so what I want to do now is introduce you to the structures of each subunit and then how they go together and what I think you need to think about as spectroscopists trying to study intermediates in Metallo-cofactor chemistry. So here, the large subunit you need to remember is called alpha 2. This is a homodimer in dark blue and light blue. It has 3 nucleotide binding sites. So it has the C site, the catalytic site, binds 4 substrates. I mean, how do you control all of this? It has the specificity site, the S site. It binds 4 deoxynucleotides. What deoxynucleotide binds here controls which substrate gets reduced. So if you want to reduce GDP, you need to have TTP. If you want to reduce ADP, if you want to reduce ADP, you have to have GGTP. Anyhow, there's a set of rules, and it's pretty much conserved through all classes of RNRs, which I think is amazing. And the third nucleotide binding site, which I'm going to talk about a little bit, and I think was underappreciated, is the ATP cone domain. It's the N terminus, and it's circled in green. It binds ATP or DATP. DATP inhibits all class 1A RNRs that we know about, and ATP activates. So the ratio of ATP and DATP turns the enzyme off and on. Okay, so what's amazing about this? In this case, you can have 3 different binding sites in each subunit full of nucleotides, and you get no chemistry. Why? Because we haven't generated the sulfur radical yet. Okay, so it's just sitting there loaded depending on deoxynucleotide pools. Okay, so the second subunit is beta 2. It's also a homodimer. You can't see the little irons there, but deeply buried in this subunit is the diferric iron cluster, whose function it is, and we'll talk about at the end, starting with iron and oxygen, is to oxidize tyrosine 122 to the tyrosyl radical. The other piece of information that's going to be key is, and this is an issue with all RNRs, a structural issue, the C-terminal tail is missing, disordered, and so we're missing 35 amino acids. Okay, this C-terminal tail, if you cut it off, you have no interaction between the subunits, so it's playing a really pretty key role. And furthermore, you'll see in a minute, there's another tyrosine in this tail that's essential in some way for this weirdo oxidation across subunit interfaces. Okay, so here's the model, this model, and let me say this at the outset. I started out putting in references to everybody, and after I had 15 pages of references and had to start giving people credit all along the way, I decided I couldn't do it. Then I asked myself, so I'm digressing again, not good. So then I asked myself at the end, should I start with my collaborators? So let me say this now, depending on how far I get. Really, I couldn't have done anything without all my collaborators. If you look at it, you know, I collaborated with spectacular structural people, spectacular spectroscopists, people that know how to do electrochemistry, people that taught me how to synthesize nucleotides. It worked because of the team of people. So nobody get offended if I left your name off the slide, okay? I did my best. And I'm also over 70, so I can't remember anything anymore. All right, so I have an excuse. All right. So really, Ullin and Eklund proposed a model for the active form of ribonucleotide reductase, which you see in all the textbooks. So here's alpha 2. Here's where the C site, the substrate site. Here's the specificity site in another view, which controls through loops which substrate is reduced. And in this site is that cysteine on the top face that needs to be oxidized. Okay. Here's the second subunit, also a homodimer. There are a couple things I want to point out here. So the homodimer, here's the tyrosyl radical. We can't see the C-terminal tail. We're missing 341 to 375. And when they, in one crystallization effort, they had a peptide to the C-terminal tail, 360 to 375. You can see that, but you're still missing 20 amino acids, and that key tyrosine, this key tyrosine here is missing. So we're missing a big chunk, and that's part of our problem. Okay. So if you look at this model then, and I think we have strong evidence I'll show you for this model now. This tyrosyl radical in the docking model, based on shape complementarity, is located 35 angstroms from the cysteine. So how do you do an oxidation? You got to use your imagination. There are no other metals except for this metal. And so it was proposed by Ullin and Eklund that you were going to involve a series of redox active amino acids. And while that's mostly what we've worked on, I'm not going to have very much to say about that today. But this is like the most amazing reaction in biology, in my opinion. Anyhow, I'm very opinionated. So anyhow, the other thing I wanted to say here, and this is really important, is that in all the textbooks you see that this is a symmetrical alpha-2, beta-2 interaction, and in fact it's not symmetrical. So it's asymmetrical. Chemistry only happens on one alpha-beta interface, and you'll see evidence from mechanism-based inhibitors and unnatural amino acids. Every single one of them tells us about asymmetry, okay? So this is the reaction I'm not going to be talking about today. I just want to introduce you to it because it's so different from the other ways that nature has chosen to oxidize sulfur to a cysteine radical. So you can put these two proteins together, nothing happens. This tyrosyl radical sits there in E. coli for 4 days. Amazing, given it's dying to be reduced, okay? It's only when you bind substrate and effector over here that you change the conformation of the protein. You need both of them. And you uncouple, conformationally you uncouple the system. And so you get rapid oxidation, rapid reduction, and rapid back electron transfer on every step. So all the chemistry, this long-range oxidation, the chemistry of nucleotide reduction, and a back electron transfer is completely masked by a physical step. This happens quite often in regulatory systems. So to study this, we have to perturb it, and then how do you go from the perturbation back to the wild type? That's what you face when you're an enzymologist. Okay, so the other issue that so here's the 356 at the interface we can't see. So just let me show you what our working model is. So if you want to oxidize tyrosine, you not only need to lose an electron, you need to lose a proton. So we've been focused on, you heard about this in the last talk, proton-coupled electron transfer. Electrons can tunnel very fast, 10 to the 8th per second, over long distances, 10, 15 angstroms. Protons are much, much, much bigger, mass 2,000 times bigger. They have to be precisely aligned to have the proton coupled to the electron transfer. This is different from almost all the other systems out there. So the model is the following, and this is a figment of our imagination. This is the RNR Stubbe model, but the fact is that we have a lot of data that supports it. So substrate and effector bind, you get a conformational change, and then the chemistry goes like a bat out of hell. Okay, so you get proton transfer, electron transfer. This is a hypothesis. Proton transfer, electron transfer. We need to go across the interface we can't see. We now have collinear proton coupled electron transfer till we finally get over here to generate that cysteine on the top face. Okay, now we do nucleotide reduction. Okay, that's also fast. And then we have to go all the way back. We know from studies I'm not going to talk about today that electron transfer in this region occurs at 14,000 per second. We know that nucleotide reduction occurs at 150 to 200 per second. So everything is conformationally gated. And really, to figure out how this thing works, you need to think about the dynamics. Okay, so these are the issues, and these issues have plagued everybody's life in the RNR field. This is the putative alpha-2, beta-2 is the active species, which I believe it actually is. And so what do we know? We know that the subunit interactions are weak inside the cell. The concentrations are low. 0.4 micromolar. We also know from studies in the last few years that beta-2 can act catalytically. So one beta-2 can serve as 5 alpha-2s that can come off and on. That means the interaction between these two is dynamic, in contrast to the pictures you see. And so recently our focus, in collaboration with the Drennan lab and the Asturias labs, a cryo-EM person, we've really been trying to figure out how we can trap this in one state to get the missing pieces of structure, and then eventually use this information to understand the dynamics, which is the way it works. So we were really excited when we made this construct. I'm not going to go into it, but I'll show you again. We can put an unnatural amino acid in this and a mutant. And the two subunits stick together more tightly. And this is a negative stain EM at 25 angstroms. And for the first time, we can see the asymmetry. We can see the half sites reactivity. Now, of course, how does that relate to the wild type is what we're actively trying to figure out. Okay, so how we tried to perturb the system so we could understand what was going on from a chemical perspective. And so one of our approaches has been to use site-specific incorporation of unnatural amino acids, which we did initially in the collaboration with the Schultz lab. Let me just say this, if we don't get to the end, it's all right, okay? So let me just say this, because you're young people trying to figure this out. We spent 15 years with Peter doing experiments, trying to get unnatural amino acids in at a scale where we could make 100 milligram quantities of protein, okay? We couldn't do it, and we didn't publish anything. But now it works, and it's opened up really things that we could never have dreamed we could have done. So I think persistence and passion are key things you want to remember when you're a young scientist. Anyhow, we've been able to site-specifically incorporate all of these amino acids into any position in that pathway I showed you. I wanted to, let's see. This is going to take time too, because I can't remember what to do. All right, let me just, how about this? All right, so you remember, these are just little helpers because it's so complicated, nobody can remember anything. Okay, so this guy you're going to see again, aminotyrosine. We just measured for the first time the formal reduction potential. It's 550 millivolts, easier to oxidize than tyrosine, completely different from what's in the literature. And here we have fluorinated tyrosines, where we can modulate the redox potential over 160 millivolts. And that's enough to allow us to study and uncouple the conformational gating in this pathway here. Okay, so I want to talk about structure. That's what I'm in the middle of talking about now. We've used two methods to look at structure: electron microscopy and pulsed electron electrode double resonance spectroscopy. And then what you're going to see for the rest of my talk is we've used, and this is where I've collaborated with every single spectacular EPR specialist in the country and in Europe. Okay, so there are a lot of people who made this possible. Anyhow, and what's the other thing that's amazing, you should know that. You take this all for granted. But, you know, it took us to get a 140 gigahertz EPR spectrum in Bob Griffin's lab when I first moved to MIT. It took us 3 years. Okay, now you guys just drop it in, you give it to someone, they give it back. And furthermore, we couldn't simulate anything. Okay, so nowadays, if you look at the simulations, it's mind-boggling. So you guys are in the right age to have fun doing science, and you can answer questions I couldn't dream of addressing or answering back when I was your age. So anyhow, we use these methods. You'll see a little bit about this. We use transient absorption spectroscopy. We made a photoactive ribonucleotide reductase, which allows us to do things from the microsecond time scale to the millisecond time scale. And we use those as probes to study what's going on in nucleotide reduction and in cofactor assembly, which is what I'm going to be talking about today. So let me just show you one thing that was completely unexpected, but I decided we really had to know how these subunits were going together, and I'll tell you why I think that's important to bioinorganic chemists. Okay, so what we found out by accident, so here's the beta subunit, here's the alpha subunit. You bind, without substrate and effector, these guys sit here. Once you bind substrate and effector, you get your conformational change. This guy gets reduced, and you form an amino tyrosyl radical. This guy is a thermodynamic hole, so it's dead. So you can go from here to here. And what I'm going to show you, I'm not going to show you all the details, but when you do that, something happens to the conformation of the protein, so that instead of having weak interactions, the interaction changes by a factor of 10 to the fourth. Okay, so something happens when you get into the radical transfer chain. Okay, here's another example where we put fluorinated tyrosyl radicals. This is in the position of the normal tyrosine. It uncouples the conformational gating. We've studied this in a lot of detail. I'm not going to go into this. But what can happen when only when substrate and effector are bound, you trigger off electron transfer. You make a deoxynucleotide, you do back electron transfer, you generate this radical in the pathway, which then can slowly reoxidize this tyrosine. But the point here is when you do that, when you put another radical in a different place in the pathway, the subunits stick together again. So that's where our basis for trying to figure out how are we going to get a picture of the interaction between the two subunits. So this just shows you another way we've been looking at this is, this is the EM way. So in this way, here's the aminotyrosine. You have two tyrosines. This is alpha beta. When you add substrate effector, this gets reduced, this gets oxidized. They stick together, and the enzyme's dead. It can't go any further. And so what you have then is a diagonal distance between this aminotyrosine and this alpha beta, and this tyrosine and this alpha beta. And that we're going to use to do Peldor, and it also made the two subunits stick together. And so here's what we observed experimentally. We were incredibly ecstatic. This is negative stain EM. And this is 2D classification, and then you can use the 2D classification to reformulate the 3D structure. And we can fit it with alpha 2 and beta 2. And that's the first evidence that this docking model, which has been around for many, many years, is in fact correct. If you use Peldor, I understand you talked about this, pulsed electron double resonance methods that allows you to measure distances between two radical species, tyrosyl radical and another radical we're going to generate there that have weak dipolar interactions. And so the idea was here, you can measure this distance. And what you can do on this side, we're going to replace this tyrosine with aminotyrosine. And then in the active site, we use a mechanism-based inhibitor where we generate a radical. And so what we can do then is we add the nucleotide, this gets reduced, and these guys all get oxidized. We do 5 separate experiments, and then you can measure distances. And we have a docking model, and you can ask the question, does the docking model fit with the distances? And so we have more distance constraints for how the two subunits go together. Okay, so we tried everything. I collaborated with many crystallographers trying to get a structure of this protein. And the reason we can't is it's moving around. Okay, so we then turn to the inhibited state, which all RNRs 1As are inhibited by DATP. And DATP binds that green cone domain I told you about before. And so we were pretty desperate. And when we did this, so this is the active model of the complex. There's the tail that involved with holding the two subunits together. And when we got DATP, the first thing we did was negative stain EM, and look what we saw. Like, this was like mind-boggling to me. So here we have alpha 2, beta 2, alpha 2, beta 2, with a hole in the middle. Okay, so here's alpha 2, beta 2. So why does this inhibit? It inhibits because the distance between this tyrosyl radical and this cysteine, which is 35 angstroms, is now changed to 60 angstroms, and you no longer have this tightly coordinated pathway to get the electron transfer. So this inhibition by the ATP is working by preventing the long-range oxidation, if you weren't a believer in that process. And just to make a point, So here's the green domain. This is the ATP codomain interacting with beta. Here you can see a little green interacting with beta. I also want to point out that the nice thing about EM is you get thousands of particles, hundreds of thousands of particles. And this isn't, like people draw it, a rigid system, right? It's got to be flexible because the thing has to go back and forth, and you have to have dynamics. ATP needs to turn this back into the active state. And remember, we have a combinatorial library of nucleotides inside the cell that we don't get. That's what you guys need to figure out so we can think about it better. Okay. So I just want to make one other point, because I think this is mind-boggling. We focus on the human enzyme as well. The human enzyme, alpha and beta, is structurally homologous to the E. coli enzyme that I just showed you. Okay, so, but its mode of inactivation by DATP is distinct. Okay, so here's alpha 2. And if you throw in DATP, what do you see? So you see alpha 2, alpha 2, alpha 2. It's a trimer of dimers. And what you see, these little green domains, that's the ATP cone domain. You just saw the ATP cone domain interacting with beta. In the previous slide here, we have no beta, and it's formed a hexameric state. And we believe that the mode of inhibition is related to the fact that we had 3 betas in one EM experiment we did. We only saw 1, it was all over the place. And from other studies we've done, we believe that this is inhibited because this complex is tight enough that beta 2 cannot get in to form an alpha 2 beta 2 complex. So ATP also can make a hexamer, but it's the dynamics of the hexamer that's key to the way it functions. Okay, so let me just now say something and summarize, and why I think this is important to people doing bioinorganic chemistry. And I think we were exceedingly lucky that our experiments worked, because we had no knowledge of this when we did a lot of our experiments. First of all, we have alpha 2, beta 2, alpha 4, beta 4, alpha 6, alpha 4, and recently discovered by Bo Lando in my lab, a non-canonical alpha 2. These are all structurally the same, okay, but they're forming different quaternary structures. The issue is inside the cell, and we've studied like alpha 6 inside the cell, and we know it happens inside the cell in the case of the human enzyme. Okay, so you can imagine if you're doing a stop flow experiment at micromolar, and you're doing a Mossbauer experiment at millimolar, the concentrations are pretty different. So you always worry about the kinetics and how they change, but one way you could change all of this is by forcing alpha 2, beta 2 to go into alpha 4, beta 4, and I think this explains a lot of the kinetics I did 25 years ago on mechanism-based inhibitors, that we had an equilibrium between these species. So I think you need to think about it. If you're working on a little rock of a protein, it's not a problem. If you're working on something that's regulatory, it's likely to be a problem. So again, RNR, the quaternary structures are nucleotide dependent in ways we don't really get. And again, we have distinct concentration dependence, which just means in the back of your mind you need to think about this issue. Okay, so here's the dreaded mechanism of ribonucleotide reduction. This is how I got tenure. I love this mechanism. Frank loves his electronic structures, and he loves computations. I love organic chemistry mechanisms. Okay, anyhow, nobody believed this mechanism. But I'll give you, so let me just tell you a few points. We're not going to spend much time on it. So here's the nucleotide. Here's the deoxynucleotide. Here are the 2 cysteines that get oxidized to provide the reducing equivalents, okay? And here's that sulfur radical I just showed you about that needs to be generated that initiates this process, okay? So what I want to do then is just show you a few steps in this reaction and then make a few points about how we went about studying this, because I don't really think it's any different than studying a diferrous cluster in oxygen going to a diferric tyrosine radical cofactor. I think the way you approach mechanistic studies are exactly the same, and the caveat is you can never prove a mechanism, and your mechanism is always has pot shots taken at it. That's what science is all about. What you want is the truth. Okay, so keep that in the back of your mind. Okay, so we're going to pull off the hydrogen atom, and one of the tools is going to be a mechanism-based inhibitor. All of our mechanism-based inhibitors that means that they pull off a hydrogen atom, okay? And then once you do that, you lose water. That's an irreversible step. And then somehow, you need to do redox chemistry. I think this part of the mechanism is pretty well understood. The redox chemistry is less well understood, but I believe, I won't have time to talk about it, it also involves sulfur radical-based chemistry. Okay, I think sulfur radical-based chemistry is in a lot of places underappreciated because it's really hard to see it, and RNR turned out to be, we turned out to be lucky to be able to see intermediates. Okay, so how do we approach this? Let's see. Yeah, how do we approach this? So we use three approaches, standard approaches, but you use this when you study iron oxo-mediated oxidation of something. You look for an isotope effect on the reaction. Okay, so we saw no Vmax isotope effects, only V over K. From our photo-RNRs, we can measure the rate constant for this process because the photo-RNRs uncouple the conformational gating of 14,000 per second. The turnover of the enzyme is 5 per second. Okay, that just shows you the physical step is really important, or steps are really important. You all use site-directed mutagenesis. We use site-directed mutagenesis. You're going to see this later on. And I just told you we use mechanism-based inhibitors. And they're all 2' substituted derivatives. And I'm only going to talk about one of these, which is widely used by the bioinorganic community. And I'll show you why. But all of the studies we've done on mechanism-based inhibitors were central to figuring out what the heck is going on over here. We spent a lot of time studying this. So here are the mechanism-based inhibitors. Again, I'm going to specifically talk about the azido compound. I just point out that gemcitabine is a drug that's used clinically. And it is a substoichiometric mechanism-based inhibitor of ribonucleotide reductase. One equivalent per alpha-2 is enough to kill the enzyme. Interesting. And the products we see in the test tube are like the products we see in cancer cells. Okay, so what's happening in these reactions? I'm not going to go into it in detail, but they're mechanism-based simply because you remove a hydrogen atom and then you need to lose water. Or here you lose X. And now what happens, depending on what X is, the protonation states of the groups on the top phase and the bottom phase are different, and different chemistry happens. And it's complicated, okay? And I'm not going to go into any details, but if you ever get to something like this, most of you who know a little bit of organic chemistry realize this is unstable and the whole thing falls apart. And that's how you end up inactivating the enzyme. You can alkylate the enzyme with a highly reactive furanone. Okay, so what happens with the azido CDP and why, or ADP or UDP, and why am I talking about it? So N dot, okay, has been widely used by most people studying Metallo-cofactor assembly and interaction with the second subunit of new ribonucleotide reductases as an indicator that you're actually doing chemistry in the active site. Okay, so what do we know? This was discovered by Senglander and Eckstein in 1976. We studied am I on? Yeah. Okay. Okay, so the tyrosyl radical goes away, and this is 5 to 10 minutes, so this is slow. And what do you see? You see an N dot. Okay, N dot. How do we get, what is this N dot thing? Okay, so if you let it sit around longer and you isotopically label it, you can isolate the pieces. Okay, so the whole thing blows apart, basically, and that's what happens with the mechanism-based inhibitors, including we get nitrogen gas release. That's pretty amazing. Anyhow, what I want to just show you is what the N dot is. So it only took us and our collaborators 10 years to figure that out. And so how do we figure this out? You know, we labeled the 1', 2', 3', and 4 prime positions with deuterium. We labeled the base with N15. We labeled the azide with N15, the whole thing and the single nitrogen. And we labeled the oxygen with O17, okay? And the structure of N dot is this. So in addition, this is the protein, and this is one of the cysteines on the bottom face. We deuterium labeled the cysteine. Okay, and I was going to show you the spectra. This spectrum, it took us a long time to get, but I'm not going to show you the spectrum. We did many, many experiments, and this is the structure. And it makes a point that I want to hammer home. First of all, none of the things we've done on RNR could we have ever done if we were afraid to make molecules. So you saw that in the previous slide. Making molecules was the key to everything. And I learned that from a number of nucleotide chemists who were very patient with me. So we had to isotopically label everything. The second thing is that if you're studying a reaction, you don't let it sit around for minutes. You want to quench it early. Because if you quench it on the minute time scale, in most cases, you have chemistry going on in the cavity of your protein. And that cavity is not reproducible with respect to what you can do in the test tube. It's constrained the chemistry that can happen. So you can get something weird like I just showed you on the previous slide, which is actually intellectually informative, but it took us a long time to figure it out. And then the other thing I want to point out is if you're looking at this, you must do spin quantitation. The azido UDP or CDP is a half site reactivity. One equivalent kills alpha 2 again. And if you're only looking at 0.1 radicals as opposed to 0.5 radicals, you have to make the decision, are you going to chase this down or not? And I would say not, anyhow. Okay, so I wanted to say a few more things. And I wanted to say this is an important experiment, and it does bring back metal-based reactions. Okay, so I've showed you an S dot, and nobody's seen S dots. How do we know that this is really an S dot? Okay, and I also believe now that a lot of thiol radical-mediated chemistry is happening on the bottom face in the reduction process as well. But I'm going to really focus on this S dot. Okay, so we just saw that the S dot, whoops. Okay, the S dot could come from here, but we saw there were other metallo-cofactors that could generate the S dot. And so the system where we found the S dot spectroscopically is the class 2 ribonucleotide reductase. So the class 2 ribonucleotide reductase is really amusing. It's a monomer. All the others have complex quaternary structure. Here's alpha 2. And all I wanted to show here, I just grabbed this fast because I couldn't find a good structure. But here's where the catalytic site is, and the specificity site, which governs which substrate is reduced, is formed by the 4-helix bundle, 2 helices and 2 helices from each monomer, and that's where your effectors bind, okay? So, but this is a monomer, okay? And if you look at the monomer, what has nature done to retain the specificity site? It's a monomer, and here, the specificity site is built from the 2 monomers. So what she did, I think this is amazing, is she grafted on a 2-helix bundle. That's what we isolated out. This is Lactobacillus leishmanii. And so this is how she does the same specificity of reduction as the class 1A ribonucleotide reductase. This was done by Mike Sinchek when I first moved to MIT with Kathy in 2002. Okay, so this enzyme uses B12. Okay, so if you think about B12, we've known a lot about B12. And we know from many people's work, Abel's and Aragoni, whatever, that these enzymes can undergo homolysis of the carbon-cobalt bond to generate cobalt-2, which is EPR active, as a spin of a half. That's going to become important. And this putative 5' deoxyadenylyl radical. And the question then you can ask yourself is that could S dot be generated by the 5' deoxyadenylyl radical? Okay, so whoops, getting completely mixed up here. Okay, so we did an experiment, and this to me was one of my favorite experiments that I've done in my career. This was done by Licht. And so we took our 3' tritiated molecule. Remember the 3' carbon-hydrogen bond cleavage. We got cleavage. Then you would expect this to pull up a hydrogen atom. You then do the chemistry. You convert nucleotide to deoxynucleotide. In the end, if you pulled off a tritium, you can at this position you can put back either hydrogen or a tritium. You select against tritium. Hydrogen, you have tritium in the 5' deoxydenylyl radical, which can reform the carbon-cobalt bond. We see no tritium in the cofactor. Okay, so that mechanism, the paradigm for B12-dependent reactions, was not correct. And in fact, early studies by Abels and Beck, actually, and I had suggested that when I finally went back and read the literature in the 1960s. If you take tritiated water, and what I forgot to tell you was the B12 enzyme can not only catalyze the carbon-cobalt bond to make deoxynucleotides, in the presence of the effector only, it can also cleave the carbon-cobalt bond. So you don't need to have the substrate around. So we have two ways of looking at what the B12 cofactor does. So here what happens is you get cleavage. Here we use the effector. And so the hydrogen from cysteine or any monoprotic base can exchange. We then cleave the carbon-cobalt bond. We remove a hydrogen atom here, and then we reform. In the end, we can put the hydrogen back on, and again, we select against tritium, and we reform the carbon-cobalt bond. Is that my?
It might be your fault.
So normally I never hear it. People call me all the time, and I'm so deaf I can't hear anything. Does somebody? I get all these robocalls. Does anybody know how to turn those off? Yeah, sorry. I'm going to get rid of. I hate phones. Sorry. Anyhow, I didn't want to get distracted. I did get distracted. But the fact is that we start over here and we see, the question is, do you get tritium in the cofactor? And the answer is you do. And there's only one way you can do that, is that if you have an exchangeable base and that you can undergo this reaction. Okay, so what is the evidence of that? I was completely convinced at this stage that the class 1A and the class 2 had similar mechanisms, and they were going to involve a sulfur radical for the following reasons. Okay, so here's the intermediate. You can get exchange reaction, or you can get nucleotide reduction reaction. This cleavage of the carbon-cobalt bond, 250 per second, it's fast. Deoxynucleotide formation is 50 per second, it's 150 per second in the 1A. But the rate limiting step in the steady state is re-reduction of the disulfide to sulfhydryl groups. And so we set up and did an experiment. We've done them with both effector only and with the effector and the substrate, and this is what we saw. And this spectrum was first reported by OJ, one of my former colleagues at both Wisconsin, actually, and at MIT. And this is what they observed. It doesn't look like cobalt-2. For those of you who know what cobalt-2 looks like, it's EPR. This is a spectrum. And these little features here that you see are due to the nuclear. Cobalt has a nuclear spin of 7/2, so these are hyperfine associated with the nucleus, but it doesn't look like cobalt-2. So if you generate a sulfur radical in cobalt-2, they're both paramagnetic. They can have dipolar interactions. They come together. They can get exchanged coupled. And so, and when they did this experiment, they had no idea really what the structure of this was. And so we thought because of the exchange reaction, it had to be a monoprotic amino acid. And so we deuterium labeled all the cysteines and RT-PR. And when you deuterium label, now you have deuterium labeled S dot and cobalt-2. And you can see marked sharpening of the hyperfine features on the cobalt, telling you these guys are interacting. Okay, we did this in the normal substrate and with the effector. If you quantitate the spin and you compare the amount of cobalt-2 by stop flow, and you look at the spin, you get 2 spins for 1 cobalt-2, again, suggesting you have 2 radicals. And this is one of my favorite experiments. This goes back to physical organic chemistry. If you have an S dot and a C dot, and you have a hydrogen and a deuterium, based on fractionation factor analysis, the deuterium wants to be on the sulfur. And the fractionation factor is about a factor of 0.5. So we did an experiment in complete water and in D2O. And we look at cobalt-2 formation. We look at the kinetics and the thermodynamics. This is the thermodynamics. And what you can see here is this case, we have twice as much with the deuterium on the sulfur of this cobalt-2 as we do with the hydrogen on that position. So that strongly supports a sulfur radical involved in this reaction. Okay, so, all right, I'm gonna skip that. So we studied that reaction. We studied that reaction in a lot of detail. I don't have time to go over it in detail, but how does it relate? How does that observation relate to all ribonucleotide reductases? So I went back to the structure I showed you in the very beginning. All ribonucleotide reductases have the same active site. And the cofactor, or whatever mediates the oxidation, is sitting under the cloud, the 5' deoxyadenosyl radical, or in this case, the tyrosyl radical, it sits way over there, mediates this change. So it's a similarity in structures that convinced me a long time ago that all these guys had to involve sulfur radical in catalysis, but we have never seen the sulfur radical in any of these other systems. The only place it's been observed is in the B12 enzyme. Okay, all right, how am I doing? Okay, I think I'm all right. Let me just think for a second. All right, I think I'm all right, even with all the digression. All right, so let me just say 2 things. Okay, so we're back over here, and I just showed you we know where we got the sulfur radical from. What about the bottom face reduction? Okay, and we quit working on this for a while, and we came back. This is one of these things when you work on mechanism, a new tool comes out, and you go back and you re-examine it, you're thinking about new ways. And here, without going into details, I propose this weirdo disulfide radical anion, and we looked at the class 3 enzyme, which uses formate as a reductant, has a methionine and a sulfur there. But by rationally thinking about how to modify the protein, we were able to trap these radicals, okay? So by thinking about this from our mechanistic understanding, we were able to trap 2 radicals. And this one is a disulfide radical anion. No one has ever seen that in a biological system. It's chemically, it doesn't get converted into product, so it's an end product. We trap it and then that's it. But with the methionine and the cysteine, this one here, with formate in the class 3 enzyme, we saw this bizarre radical. by isotopic labeling, we isotopic labeled everything. We discovered a new thiosulfuranic radical. No one's ever seen this before. They've seen it once or twice in the EPR literature, but it's kinetically and chemically competent to make deoxynucleotide. So I think you are doing sulfur radical chemistry on the bottom face, and I think the key to RNR is the sulfur radical chemistry. Anyhow. Okay, so I want to go back now to where does this thio radical come from in the class 1? And so we're finally into the case where I can talk about some metals, or metals other than cobalt, so there's metals that are interesting to this crowd. Okay, so what do we know about this cluster? I mean, this is all built on seminal experiments from the Swedish group, Britt-Marie Schoiberg, Astrid Brosland, Peter Reichard, Mark Fontekhav. I mean, there's a list of people, Larry Kay, a lot of people have worked in this field, okay? And so what I want to do is show you what we think we understand about this cofactor and what I think the issues are and why I changed from studying this in the test tube to study it inside the cell. Okay, so the 1A has 2 iron 3, starts with 2 iron 2s, and it ends up with 2 iron 3s and a radical, okay? So it's a diferric tyrosyl radical cofactor. While studying in vivo what was going on with assembly of this cluster, we realized we had to think about another class of ribonucleotide reductase, the 1B, and that's when we got in and started studying that, and we discovered that we think now all 1Bs, as was originally proposed by Olling and Follman, many years ago, highly controversial. We now know we can make a dimanganese tyrosine radical cofactor. And as I've already shown you in the first slide, the 1Cs, which have been studied here extensively at Penn State, don't have the radical, they have a phenylalanine, but the oxidant is a high valent manganese iron system, which everybody is interested in. And so I'm interested in it too, I just don't understand them. Okay, so studying this, and we had to study this. We were forced into studying, I'm an organic chemist, we were forced into studying this because when you isolate the protein, it's apo. You don't have any metal cluster. Okay, but so it's inactive. So you have to somehow assemble a cluster. And fortunately, Atkin in the early 70s showed us that if you dump in iron 2 and oxygen, you can assemble the cluster. So that was a key observation. And then Marty spent his whole thesis trying to figure out how to do this in a reproducible way. Okay, how can we get the maximum amount of radical and cluster? Isn't that true? Yeah, so anyhow, so we look at the stoichiometry of the reaction. We believe oxygen is the oxidant. 2 irons are converted to 2 iron 3s. Tyrosine is oxidized to a radical. Oxygen is the oxidant, and so we need an extra electron to reduce the oxygen to water. So that's the stoichiometry of the reaction. That's what a chemist would do to try to understand what's going on. Okay, so that's the first thing we did, and it wasn't just us. People did this in many different systems, and we all got the same answer. Okay, so here is a cartoon overview of our original model from a long time ago, which has been extensively fleshed out. I'm not going to talk about all the details except for intermediate X, which is highly still controversial in my opinion. So anyhow, you start out with apoprotein. You got to add iron somehow. You need an oxidant. Is oxygen the oxidant? You form a peroxide. Data is from Marty's lab with Baldwin, has provided evidence peroxide with mutants. That peroxide can get reduced. This tryptophan that I have in brackets on the pathway to form a tryptophan cation radical, which we can see by absorption at 516 nanometers, and iron 3, iron 4, which when one electron reduced comes to this intermediate X. So again, here we are with a high valent iron 4, iron 3 species, and I think the one thing that we all agree on is that this species is able to oxidize tyrosine to the tyrosyl radical to generate the cofactor that makes RNR active. So really what I'm going to focus on now is what we know about intermediate X. And I'm going to raise a few issues to this crowd, okay, as sort of an outsider. Okay. So we need spectroscopic handles. You've seen this in Frank's lab. You've seen this during your whole workshop, right? What's spectacular about ribonucleotide reductase is it has a sharp absorption feature of 410. That's the tyrosyl radical. Sort of like amazing, pretty good extinction coefficient. It has an absorption feature at 390. And this stuff over here, 325 and 365, is the diferric cluster, okay? We study this by EPR. This is the tyrosyl radical. This is what we spent most of our life. I spent most of my life doing at 9 gigahertz. You can see that you have, this is a derivative spectrum, you have a doublet spectrum associated with a hyperfine interaction of one of the 2 beta hydrogens. The other one is too small in the case of our protein, and hyperfine interactions from the ring protons. And this is the 140 gigahertz spectrum, the first one, high field spectrum we got. This was many years ago, that took us 3 years. Okay, we now routinely do 263 gigahertz spectra. And what's spectacular about it is the components of the G tensor are split out, and this GX component, which is indicative really of the electrostatic environment of the electron distribution on the tyrosine ring, has been incredibly informative. So having it spread out has been incredibly useful. And we couldn't do anything without Mossbauer. And I will say this, and I should say this at the beginning. Marty went to Dale Edmondson, Vincent Wynn's lab. I even went there once. And really, they got us started on freeze, quench, EPR, and Mossbauer. And we couldn't have done anything without their patience with us to do all this chemistry. So we're grateful to them. Neither of them are here in the audience. But I do have pictures of them, but if I don't get that far. Okay, so So what I want to do, so we've used different, just like you've already seen, I think, from Frank, we use different kinds of spectroscopy to try to figure out what we're looking at. Okay, do we see anything? And if we see something, what is it? Okay, so we use stop flow, micromolar usually in our case, and that's the millisecond time scale. We have done rapid freeze quench experiments and analysis by EPR, 9, 35, and actually more recently 260 gigahertz EPR. We can do Endor. We started out with mostly Q-band, but we have now done that at high field as well. We've done even high field and low field Peldor. We can do Mossbauer, and you saw Frank's studies with MCD, which Marty did with Solomon a long time ago on RNR. The problems with these methods are they're discontinuous, and they take a lot of protein. You have to isolate grams of protein. So you really have to have control over your protein, making it and figuring out how to load it in a homogeneous form. That is not trivial. The biochemists often, like me, have often always get blown off. You know, it's trivial. It's not trivial to make homogeneous proteins with single metal clusters, which make the spectroscopy tractable. We've also used photo-initiated reactions, and I won't have time to talk about the computational methods, but in the PCET process here, we studied with Marina Bonatti without Frank's assistance and structure with the unnatural amino acids. I think we never could have simulated our Endor experiment. So this is an example where everything worked together to give us a lot of information about how this process worked. Okay, purity of your metallo proteins and metal loading. Okay, with R2, we still don't know how to load it. We see 3.4 to 3.6 irons and not 4. We never see 2 tyrosyl radicals per iron 2. We see 1.1. We have some of it loaded as a diferric tyrosyl radical cofactor. We have some of it as a diferric cluster in a ratio of 65 to 35, probably interesting. So that's an issue because that then lowers your signal to noise and it makes it more challenging to do what are already challenging experiments. You never have pure intermediates when you freeze quench something. You know, if you're lucky, something builds up and goes away, but if something is there for only a transient amount of time, the limit's 10 milliseconds in the rapid quenching, in our hands in the rapid quenching method. So you're going to have to do subtractions. And so to do that, I really think you need to have pure standards. Otherwise, the analysis of these complex kinetic experiments are really quite hard. A lot of metal clusters aren't EPR active. EPR is good because it has higher sensitivity. Mossbauer is bad. Where's Carson? I've been trying to get him to do Mossbauer. You need to do Mossbauer at micromolar. How about nanomolar? That's what I really want. Anyhow, I don't want much. But we want to do this in the cell, and millimolar is just out of control. Okay, anyhow. And they oftentimes have low extinction coefficients with iron things like Frank was talking about. You can perturb the system with mutagenesis. And in RNR system, when you perturb with mutagenesis, really all hell breaks loose quite a bit. In other words, you get a lot of reactions going on simultaneously, which makes the analysis even more challenging. And you have to isotopically label. Okay, so this is intermediate X, which Marty found when he was a graduate student. And this is the signal. It has a G value of 2. Looks like an organic radical. Okay, and so we were delighted when he did freeze quench EPR with Dale and found that we replaced the iron 56 with iron 57, he could see broadening suggested that this was a metal-based radical. What the structure was debated. We've had an extensive collaboration with the Hoffman lab. This is some early data, but we revisited it to tell us something about what the composition of X is, and I think everybody has agreed that is high spin iron 3. You can see the isotropic hyperfine couplings versus iron 4, which are anisotropic. And so I think this part of the system we agree on that X is an iron 4,3 intermediate that's kinetically competent to oxidize tyrosine to a tyrosyl radical. Okay, so we did another experiment just a long time ago to look for the distance between the ions. You've heard about XAS method, you've learned about XAS method. This was done in 1998. We did 25 experiments in this paper, and in all cases, the analysis, and I sent this, since I'm not an XAS person, I sent this paper to the best people in the country to review this paper to tell me if we were doing something incorrectly. So this is where back and forth with the community, in my opinion, is really important. So we want the truth. I don't care what the answer is. I want the truth so I can think about the structure of what we actually have. So anyhow, we had this number of 2.5. And then there were 3 more proteins that came in the literature in the next few years: a diferric peroxide in R2, a diferric peroxide in ferritin, and intermediate Q, all of which had these short distances. Okay, that's all I'm saying. Once I saw the diferric peroxide, I didn't believe this number anymore. I'm just telling you, I didn't believe it. And that's when I decided I needed to switch to looking at this stuff inside the cell. That this was too complicated for me to figure out because I didn't know how to think about the XAS data carefully enough, to be perfectly honest. Anyhow, so what's happened is, fortunately, but it took a long time, this experiment was redone by the Penn State group, Mike Green and Laura. You here, Laura? I met her in the airport. Anyhow, they remeasured this distance, and they now have a distance that's much more consistent with the computational analysis, mostly by Noodleman's lab, that the 2.5 was just too short, which was then constraining what the structure of X would end up being. Okay. So we've done all kinds of ENDOR on this. I'm not going to talk about it. It's not we, it's mostly Brian's lab. And this is our favorite structure. This structure is not the same structure Frank showed on his slides. So we have iron 3 and 4. We believe we have a hydroxyl here, and we believe we have, in addition, 2 carboxylate bridges. This structure is distinct from the ones that have been recently published. Okay, so here, everybody is really fixated on the di-mu-oxo or mu-oxo hydroxyl species. I have no skin in the game. I just want to know the truth. Okay, and so to me, you know, it is what it is, but you need to explain our experimental data. Okay, so you can't just ignore our experimental data. You need to explain to us why we're interpreting the data wrong. So I don't think we know the answer at this stage. I think X has been around. Think about this, 1998. That is a long time. And so I think we have a short iron-iron distance. I think that is resolved. What the structure is, I still don't know whether I believe this is resolved. But I think we need to discuss it as a community. And I think that's one thing that for me has been spectacular about the bioinorganic community, is I can go to a meeting and I can ask all my dumb questions, and people are willing to sit down and spend a lot of time explaining to me the physics of what's going on and how to do the experiment better. That's why this community is so good, and you guys need to take advantage of each other through your interactions at this meeting. All right, what am I on now? Oh, okay. All right, so I decided to change in 1998 because of all these issues with XS, which I didn't really know how to do better. I thought we had done the experiment well, to look at stuff what was happening inside the cell. And let me just tell you at the outset, inside the cell is even harder. Okay. I wish it wasn't true, but you heard Lil give a talk, iron sulfur cluster. Oh, here standing on, hard, very hard. They learned a lot, very hard. Okay, so the question is, how do you start out with an apoprotein? How do you load it with metal? I've showed you need an extra electron, and what is the oxidant? And the take-home message is for the class 1, I've already sort of told you this, you have to load iron 2, you need oxygen, and you need an electron. I think that's still true in vivo. But what I'm going to show you in the 1b, which is found in pathogenic organisms, you have manganese here, and the oxidant and the electron go together to form superoxide. So they're distinct. And we believe, anyhow, in the system we've studied. Okay, so we needed to develop tools to do this. And so what we did was we constructed the beta-2 subunit, that's the one with the iron center on it, and put it behind an arabinose promoter. So that means with arabinose you can turn on the thing from micromolar to millimolar. So you can control the concentration of your protein inside the cell. We've used whole cell EPR, and I forgot to tell you this, but we've studied this in E. coli, we studied it in S. cerevisiae, and we studied it in humans. I'm only focusing on E. coli today. So that's whole cell EPR. You can just cram your, depending on the organism, you cram it into the tube and you can measure the radical. Whole cell Mossbauer Carson was able, the diferric cluster that we have in our complex is sufficiently unique that you can pull it out of the mess that you see in whole cells. The beauty of yeast and bacteria is you have knockout strains of everything. So you can control all the iron uptake systems, you can control, knock out this protein that delivers an electron or that one, and you can look at the consequences inside the cell. And we have antibodies to all the different proteins. And in yeast, we frequently use permeabilized cells, so that the cells really is sort of still, everything is engaged without dilution. Okay, so our lessons on this system really came quite a bit from thinking about iron-sulfur cluster metabolism, where a lot of people are working on spectacular science. I teach about it. I love teaching about it, even though it's still extremely complex. So one lesson you learn immediately is you look with prokaryotes, you look in the operons, and you find, is there something there that's conserved? And I'll show you, we found 2 or 3 things that are conserved, which turned out to be the handle to get us into the field. We know that ferredoxins can deliver reducing equivalents, and I'm going to show you we discovered a ferredoxin in this system as well. The key thing, and I think this is the real issue, and I still really don't think we get it, when you overproduce an enzyme in a recombinant system, most of it's not folded right. It's folded. It depends on how big it is, but it has lots of conformations. And I think the slow steps are often reorganization of this apoform into a form that wants to completely bind or completely bind the metal in the right state. And so I think you have ATP-dependent heat shock proteins, or whatever you want to call them, that play a key role. And we found these in yeast that play a key role, I think, in allowing us to deliver a more homogeneous reduced state of the iron into the protein. Where does the iron come from? Now, this is a question. Joey, where does the iron come from? You better answer this fast, you won't get tenure. Anyhow, where does it come from? Okay, so this is our model. Okay, so here we have, this is the current model. With E. coli, we know less than we know about yeast, but I'll just show you what we know. Somehow we have to deliver the metal. That would be the biosynthetic pathway. We need a reducing equivalent in oxygen from the in vitro studies. We need that in vivo. And I'm going to show you we discovered a protein, YFAE, that I think can deliver the reducing equivalent. It absolutely can. Whether it delivers the iron or not, I think iron-sulfur clusters could actually deliver iron, but that's debated. Then what happens inside the cell? How do you control ribonucleotide reduction? Well, there are many, 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 many ways, but one way you could control it is by reducing the radical. If you reduce the radical, this is the met state, the diferric state, and the enzyme's inactive because it needs the radical to do this long-range radical transfer. So what happens if you get to this state, it's inactive? Do you throw the protein away? Or can you take YFAE, re-reduce the iron 3s to iron 2, and then cycle? So you have a maintenance pathway that converts it into the back into the system. And the studies we've done in the last few years, not the last few years, 5 years ago or something, I think support this kind of a model. But the experiments are hard, and you need to read and think about them and not just believe me. Okay, so YFAE we found in an operon. We had to fold it. That happens with a lot of iron-sulfur cluster proteins. Have we folded it correctly? I hope so. But we got really clean, oxidized, and reduced state. And then what we could do, this is in the test tube now, we're not in vivo yet. We could take the met form of beta-2, so that's where the 2 irons are in the +3 state, no tyrosyl radical. And what you can do is titrate in YFAE in the reduced state. Okay, so it should be reducing the +3 state to the +2 state, and then add oxygen and see what you get. And so what you see that we can do this titration, and what we get is, look at that guy. Does anybody know what that is? The tyrosyl radical. So even in this whole mess, this lovely sharp feature at 410 nanometers immediately tells you you have a tyrosyl radical. And then you can quantitate all of this. And when we do the quantitation, we only need 2 irons per tyrosyl radical, suggesting again that the extra reducing equivalent is probably coming from the tryptophan in the protein. And if you do the quantitation, now instead of seeing 1.1 tyrosyl radicals per beta 2, we're up to 1.7. And studies we've done in yeast are close, much closer to 2. So I think there should be 1 tyrosyl radical in each beta from the studies we've done in vivo, which we don't see in vitro. Okay, so we've done whole cell EPR and whole cell Mossbauer. And this just shows you we can see only E. coli is the worst of all the organisms we've looked at. We have to go to 100 micromolar to be able to see the EPR signal, whereas yeast, we can see it at much, much lower concentrations for whatever reason. The red is the whole cell spectrum of the tyrosyl radical. You can subtract out a tyrosyl radical and you get this black thing left. And if you do a control, the control being iron species inside the cell, you get a blank line. So you can actually see this inside the cell. If you look at under conditions where, look at this, we've made 3 millimolar beta, 3 millimolar, okay? And under those conditions, 76% of the iron in the cell is in the diferric cluster. And we can tell it's diferric cluster because the isomer shifts are unique. So that's amazing. So somehow in this organism, it knows it wants to make ribonucleotide reduction. It knows it wants to convert the iron into the center because it needs to make deoxynucleotide. And it keeps trying to do it even though it can't do it. And so I think that's a comment on where RNR stands in the pecking order inside the cell. And finally, we've done this experiment where we, whoops, it's wrong. I went the wrong way. Okay, this experiment where we take top 10 cells, so this is crude cell extracts, add back YFAE, and we titrate in YFAE. And under these conditions, we get 1 tyrosyl radical per beta. So I think inside the cell that we're loading both of these centers and that the iron centers are more homogeneous. What do you do with this information? Hopefully one of you guys in the audience can figure that out. Okay. So we have all these mutants of E. coli where we could potentially knock out the iron uptake systems. Could we watch the iron move around inside the cell? And after doing a lot of thinking about this, I realized what a stupid idea. Okay, and the reason it's not a good idea is because almost all bacteria have multiple ribonucleotide reductases. So the 1A is the one we've been talking about. We can see that. That's the guy that does DNA replication. This one over here is the class 3 that uses the glycyl radical that turns on under anaerobic conditions. But in E. coli, you also have a 1B. Okay, and so what's happening when you knock down the iron uptake or you put it under oxidative stress, this guy gets elevated. So then I realized, oh my gosh, we don't know anything about 1B. 1B was extremely controversial. I won't go into the reasons for this, but Joey decided to study 1B. And we immediately saw, looking at operons of nerd E, so that's alpha, and this one's beta, nerd F is beta. We saw a nerd I and we saw a nerd H. Nerd H cycles ribonucleotide reduction. You need to re-reduce on every turnover to get multiple turnovers. Nerd I is a flavodoxin. Was annotated as a flavodoxin. But we tried to study nerd I, and so did other groups. And we looked out, and we were able to reconstitute this protein with a flavin at very high levels. And what we immediately found out is it doesn't behave. This is a normal flavodoxin. Nerd I has very different redox properties than a normal flavodoxin. So what do flavodoxins do? They can take the reduced form, this is a hydroquinone form, and they can be oxidized with oxygen gas. to form superoxide in a semiquinone radical. They can be oxidized again to form the oxidized form of the flavin, or they can be oxidized by 2 electrons. Very complicated. And I'm going to summarize our results, but for those of you who want to believe this, you need to read and think about the paper, which is complicated. Okay, so this was like, this was a eureka moment. I have 3 or 4 of these during my career where I can feel the adrenaline coming, and I get so excited. I still get excited about this. Anyhow, we took the protein, and it has 2 manganese 2s in it. We dumped in the reduced form, the nerd-I, hydroquinone, and oxygen, and then we just took a spectrum. And look what we saw. So most of this stuff is oxidized flavin, which has a huge extinction coefficient. It's bright yellow. But look at this guy. That's it. That's the tyrosine radical. So people had proposed this for decades, but no one had ever seen a manganese tyrosine radical cofactor. And that was it. Joey, did you get excited? Are you here? Anyhow.
Yes.
Okay. He was.
You didn't believe it.
Great. Anyhow, we now looked at many of these proteins. So I'm only going to talk about the ones we've looked at. The key to looking at these things are having the right reductant, or you don't get good activity. Okay, so you need to figure that out. That's not trivial. And what we found, and others have reproduced this in other organisms, is that you can assemble a dimanganese tyrosine radical cofactor, but you can also self-assemble a di-iron tyrosine radical cofactor. And that's why for years, everybody thought the 1B was a di-iron tyrosine radical cofactor. Okay, but if you look at that in all cases, if you look at the activity of a manganese-loaded compared to the iron-loaded is always 5 to 10 times higher, and we've now looked at 2 other organisms and see the same trend. So the question then is, what happens inside the cell? Is manganese interesting inside the cell, and is it what's doing ribonucleotide reduction? Has this 1B now turned in to an enzyme involved in DNA replication and repair? Okay, I'm going to skip this. Anybody that wants to look at these, you can talk to Amy. Amy and Amy. Okay, but this is an amazing result. I'm sorry I'm skipping this because this is also really exciting. So they put the nerd-I together with the nerd-F with a manganese load, and look what they did. It like took 2 hours or something, and it crystallized. And so here's the flavin, and here's the manganese. Okay, so they got a cluster of these 2 things together. And if you look at this cluster, if you look at a blow-up of this, whoops, what you see is, so here's the flavin, this is nerd-I, here's the manganese cluster, manganese 2. There's a channel of waters that go into manganese 2, and there's a channel, although there's constriction in this channel, so that the oxidant generated by the nerd-I species, which we believe is superoxide from the studies I'll very briefly show you, can channel into the active site. So you cannot take manganese-loaded nerd-F and add superoxide and get any tyrosine radical cofactor. It requires the nerd-I to be able to do that. Okay. So we asked ourselves a question, in the organism without, most of the things we do are recombinant, what happens in the host organism? So in the host organism, RNRs, they are very low levels, so sometimes you have to purify 50,000-fold. Anyhow, Ohling's lab, who was a proponent of manganese RNRs for decades, since 1988 when he was in Fulman's lab, isolated the nerd-F from C. ammoniagenes, and we isolated it from both B settles and E. coli, and you can see it's a manganese-loaded dimanganese 3 tyrosine radical cofactor. Okay, so we have studied, just like I showed you with the assembly of the diferric tyrosine radical cofactor in the 1A, we've done the same thing with the manganese cofactor, okay? I'm not going to talk about this in detail. It's been published here, but I want to point out 2 things. And again, I point out that the flavin and the semiquinone, the tyrosine radical, manganese 2, manganese 3, manganese 3, manganese 4 are all EPR active. So the analysis and reduced flavins want to be oxidized. So this is a hard set of experiments. But we convinced ourselves that nerd I can channel superoxide into the active site. That was the oxidant. So instead of having oxygen and electron, nerd I supplies the oxygen and the electron. And what we saw doing these experiments, and this again is a collaboration, we saw a manganese 3, manganese 4, we've seen iron 3, iron 4, we've seen manganese 4, iron 3, and that it is kinetically competent to generate the tyrosyl radical. And those are just the spectra that we obtained and published in that paper. Okay, so in the last few minutes, I want to ask another question. And I think this is, again, where science is headed. Is this manganese cofactor really important inside a pathogenic organism? So I set up a collaboration with Kitten, who's at University of West Virginia, because he had reported that in S. sanguinis, so this you find in your mouth, this is a bacteria you find in your mouth that if you ever go to the dentist and they're going to give you surgery, they almost always give you antibiotics because this bacteria likes to colonize in the heart and often is associated with endocarditis. Okay, so you would like to prevent that from happening, and that's how he got interested in this system. And what he showed was the manganese transporter, called SSAB, is a virulence factor. If you knock it out, the organism can no longer grow. It has a 1,000-fold effect. So we thought then, we looked at the two proteins, we isolated nerd F, nerd I, all the reductants, etc. It looks strikingly similar to the E. coli system, including the channel into the active site. And we decided we could do an experiment in an animal model that would test whether an iron-loaded tyrosyl radical or a manganese-loaded tyrosyl radical. Remember, they're both active. One is much more active than the other. We could figure out which one would be active in the animal model. So the experiment is pretty simple. In the case of the nerd D ribonucleotide reductase of class 3, if you have oxygen around, the protein gets cleaved into two pieces, so it's dead. Okay, so if you have any oxygen around, you don't get any ribonucleotide reductase activity. In the case of the iron 2 loaded protein, you can rapidly assemble. We saw that in the first part of this, the diferric tyrosyl radical cofactor. In the case of the manganese loaded nerd F, you need oxygen and nerd I. So the experiments would be simple. You really just need to construct strains that lack nerd I or nerd D. And that's what the Kitten lab does for a living. So they constructed strains where this means knockout. So you knock out either the anaerobic RNR or you knock out nerd I or E and F. Those are the two genes that are for ribonucleotide reductase. So you have wild type. And so then you grow under anaerobic. This is just straight bacteria now. We haven't gotten to the animal model yet. You grow under anaerobic conditions or you grow under oxygen conditions. Under anaerobic conditions, these bacteria are perfectly happy because they have nerd E and F that aren't affected and they can grow happily. On the other hand, under anaerobic conditions, if you have no nerd D, you can't grow at all. Okay, so if you go to 6% oxygen, and we're at high oxygen now, what happens is if you knock out the E and the F or the nerd I, all the bacteria are dead. And because you have E and F, the nerd D, the anaerobic ones, are perfectly happy. So the bacteria itself are well behaved. And so now what you can do is take those constructs and you can put it into an animal model. And so what they do is they mix the wild type organism with an antibiotic resistance marker, erythromycin, or the knockout mice where they knock out nerd I, so that's the one that makes the superoxide with kanamycin. You mix them together, you inject the rabbit, and then after a few days you pull it out, and they then count the colonies, basically, and they count them on erythromycin or kanamycin. And when they count the colonies, the results are really quite clear. And so they use 6 or 7 different mice. And what you see is that if you knock out E or F, that's the aerobic ribonucleotide reductase, you have no growth at all. That means that the organism requires that for growth in the heart, whereas these others, the wild type and the NERD, are perfectly fine. And over here, if you knock out NERD I, again, you have no growth at all. The NERD I is supplying the superoxide. And the controls show that you have high level of colony forming. So I think these studies clearly show in this one animal model that the manganese RNR can be active in this particular endocarditis animal model. Okay, so let me just close now by sort of summarizing. I've spent a lot of time talking to you about thioradicals. I think thioradicals are much more important in catalysis. I think Frank has written a really important paper on this in 1974, if I remember right, as has Simons many years ago, about how to think about looking for sulfur radicals. They're hard to find from a physical point of view. You need to think about this before you go after them. I think RNR represents a playground for controlled sulfur radical biochemistry. And I think it has evolutionary implications. Okay, so I've talked a little bit about Metallo-cofactors, probably not enough for most of you guys. We got into this because we couldn't make RNR active without the Metallo-cofactors, and I think a question of how these cofactors are assembled inside the cell and how metallation versus mismetallation is controlled is a major unsolved problem in biology. We've talked very little bit about this long-range electron transfer. I think this is one of the most amazing reactions in biology, which we've studied extensively, and I've shown you is key to the activity of RNR. I showed you in the test tube, but also the same thing happens inside cells. I haven't talked at all about therapeutics, but that's how you get funding to work on this system. RNR is the target of 4 drugs. We studied gemcitabine in detail. That's a substoichiometric mechanism-based inhibitor. Clofarabine, this is sort of amazing. Clofarabine is used clinically as well. It puts the alpha, the human alpha, into alpha-6. The nucleotide dissociates, and the alpha-6 is stable for a half a day. So somehow you form that same alpha-6 you form with the ATP, but now the conformation is stuck. This represents a new way of targeting RNR, finding small molecules that freeze quaternary structures in different states. Hydroxyurea is a drug that is used clinically. I don't know what the heck it does, but in the test tube, it kills the tyrosine radical. How that relates to what it does inside the cell, I don't know. And triopine can reduce the radical, can chelate iron. It has many effects. This is in phase 3 clinical trial. But I think RNR is a great target for antibacterial agents. And finally, I think the future is, many of you like to think about evolution. I swore I would never think about evolution till I got older than 70. I'm older than 70, so I can think about it. Anyhow, everybody is focused on the metals. I'm focused on sulfur radical chemistry, which I think happened in the prebiotic world. But I think more important than that, I think we know very little about nucleotide metabolism inside the cell. We study carbohydrates, we study fats, we've integrated this to cancer cells. People need to look at iron, and they need to look at nucleotide metabolism using all of the modern methods, all the omics methods and the gene replacement methods. And who knows what else you guys will discover. And I guarantee that ribonucleotide reductase will be fun to study for the next 20 years. So now I have to thank everybody. All right, can I I'm sorry, I have to thank people. Okay. So I hope I get everybody on here. I reorganize all these slides. But, you know, as I said in the very beginning, we couldn't have done anything without all our collaborators. So structure. So we have Kathy, my colleague at MIT, Amy Rosenzweig, Amy Bowe, Nozomi Ando. All of these guys have done structures that we couldn't have done anything without you guys. So thank you so much. We've had fun collaborating. EPR. I don't have all the EPR. I don't have Jack Pasek. I don't have John McCracken. I don't have Jim Hyde. I remember Jim Hyde from Milwaukee in the so this is like really shows how old I am. But anyhow, the EPR was Bob Gerfen. Marina Bonatti is probably been my most productive collaboration. I've collaborated with her for 20 plus years. We've published something like 15 or 20 papers. Dave Britt. Here is Vincent Wynn and Dale Edmondson, who got us into the freeze quench material. I forgot Brian in the EPR. Brian. I love Brian. So Brian is one of these guys. I ask one dumb question after another, and he's always nice to me. And you know what? But he listens to me, too. So because of the going back and forth, even though I ask dumb questions, it makes him sometimes think about things in a different way, and that's what I think is fun about science nowadays. And I think there's so much to learn, we can't master everything. This is Francisco Asturias, who's played a really key role in the cryo-EM studies. We just solved Kathy's lab, my lab, and Francisco's lab has solved an atomic resolution structure of the cryo-EM structure of the class 1 RNR. Cecilia Thomas has developed methods to measure reversible reduction potentials in a small little protein. And so we've been able to do that for all these unnatural amino acids, which is key to thinking about the thermodynamic landscape. Karsten and Marty, you know, they are the ones that filled in all the details. Marty was supposed to give, let me just tell you this. He was supposed to give the middle part of this talk, because I don't feel comfortable talking about 017 and anyhow. But he didn't get back to me, and everybody knows how nervous I am. And so then I decided, forget it, I just got to do my thing. So that's why I wrote him up in the abstract, but that's why he didn't give that part of the talk. Okay, let's see. Oh, and I need to acknowledge Mingxia Huang, who taught me a lot about yeast genetics. So we did a lot of yeast genetics in my lab, and I think yeast is a really good system to settle metal homeostasis, like you guys heard about from Lil. All right. So this is my group, and I'm not going to go through. There's my dog. She's dead. She's dead now. Oh, poor Zami. Anyhow, this is, I know, it was so sad. Anyhow, this is Kenny Kong, who's now doing we collected cryo. Yeah, I'm not supposed to talk about this, but Kathy knows that I always talk about stuff I'm not supposed to talk about. We've collected cryo-EM data now, so we will know in a couple of weeks whether the data is good enough to be able to get an atomic resolution structure of alpha and beta. Okay. You know what I forgot on the last one? I'm sorry, but this is important. That guy. Okay, everybody knows who that guy is. I didn't know anything about PCET. I couldn't have done anything without PCET, and I could have spent the hour and a half talking about our studies on this long-range radical transfer, which I think are really, really nice studies. That's the group. Ed, I need to point out Ed. He's head now. He worked with Francisco, who's the EM guy, and he came to Kathy's lab and is now the EM director of our new EM facility. These are, again, guys from my lab. Okay, these are all the guys. Here's Todd Kitten that worked on Troy. Is Troy still here? Anyhow, Troy, that worked on the manganese, understanding how the manganese tyrosine radical cofactor is formed. The EPR is really complicated. And this is something, again, I didn't talk about. I don't know why we got a line through there, but Marina Benatti and Frank were really instrumental in helping us think about proton-coupled electron transfer in that pathway that I've turned off. So thank you. I'm sorry.
JoAnne, I get two questions. So S-synchronous has, I think you said this, just a class 3 and a class 1.
It also has a class 2.
Okay.
But we've done experiments that we don't think the class 2, we've done a lot of knockout experiments of that, and we don't think that that's involved in this. We don't know what it's involved in.
Is it active? Have you ever made
We haven't ever really studied, we haven't isolated it out and studied it in detail.
My second question is about the potential of the
Right.
tyrosine. I think there was a time when it was thought to be
260.
similar or more similar to tyrosine radical.
No, it was 260 millivolts, easier to oxidize, but the number...
Now it's 500.
It's now 550. We just published a paper, and the bottom line is it undergoes rapid 2-electron oxidation. So unlike all the others where you can see the tyrosine radical formed and see it form and get re-reduced, and you have to deconvolute this through complicated electrochemistry to see the 2 1-electron oxidation potential. So it goes fast, and the people that were doing this was a long time ago. This was a guy at Ohio State whose name I'm blanking on. What's his name, Mo? Mo's gone. All right, anyhow, I can't... Clapper, yeah. So anyhow, that number is wrong. So it's really, no matter what, I think it's dead.
Okay.
So the amino is dead.
That proton coupled, or that's just a pure 2-electron
Just
oxidation?
so you can study all of that. You form the imine. You go to the imine quinone, which then can dimerize depending on how you set that up. So we did electrochemistry in Dan's lab, different kinds of electrochemistry to look at that and convince ourselves it was 2 electrons. But in the case with the little maquette, because of the differences, slight differences in the redox potential, we were able to back it out. You need to look at the detailed analysis to back out the 2 1-electron potentials,
And
which
so-
are similar, actually.
So what do you think about catechol or dopa radical being...
Yeah, so I don't know. So the dopa radical wasn't all dopa radical, dopa to dopa radical was easier to oxidize. Yeah, I don't know. I would go back and I have to go back and look at those papers carefully. But the amino, I mean, I've been worried about the amino forever. I didn't think it should be turning over. And again, one of the problems is when you put in unnatural amino acids or if you use site-directed mutants and your gene is essential, you always have low levels of contaminating wild type. Okay, that's a problem you always face. So I was convinced, although the students weren't convinced. I kept on it and on it and on it. And we finally showed, I think, in this last paper we published that it definitely is inactive. And the low levels of activity down in the low percentages are all due to contaminating wild type. Yeah. Did you stay awake, Don?
I stayed awake. The regulation is really fascinating. Can you say, do you know anything about the affinity of the effectors for the binding sites, and how does that compare to substrate binding?
Yeah, so the affinity, the ATP has 2 sites. It binds in the specificity site and it binds in the activity site. It binds much more tightly in the specificity site than it does in the activity site. The KD is around 10 micromolar in the specificity site or something, and in the activity site it's much higher, like 170 micromolar. The Km values for the substrates are on the order of depending on which substrate it is, so they have different concentrations in the side the cell, or 1 to 50 micromolar. And ATP is millimolar. So this question of ATP, DATP, and why that's the key regulatory mechanism. What is ATP doing? I think is a really, again, this goes back to nucleotide metabolism. We have ATP everywhere. I think it binds to almost all kinds of proteins and turns them off and on in ways that are subtle and plays a really important role. And I don't think we understand that. It's, there from millimolar to 3 millimolar or something like that. So this ratio of ATP and DATP, which turns it off and on, I think is really interesting.
And you also mentioned this 10,000-fold increase in affinity for the 2 substrates.
Yeah.
Do you have any idea how that is achieved?
So it doesn't, 10,000 fold is not very much. It's not very many. Hydrogen bond, if you think about a hydrogen bond, it's not that much. So our hypothesis is the following. So what triggers this long-range radical transfer when substrate and effector bind over here? And what triggers proton-coupled electron transfer that Marty and Carson and Bina, I knew I couldn't say it, so I asked him to practice, showed involved proton-coupled electron transfer. In addition to that change, things are happening at the interface. And so we looked at the interface. We have something like 1,000 sequences, and we picked out 4 residues, 2 negative and 2 positive. We did a bunch of mutants. They're all dead. Okay, so we don't see any dead mutants in RNR. They're clearly at the interface doing something interesting. And with some of our natural amino acids, they're dead with a wild type, but we can make it do electron transfer. So and that's how we got this half-sites reactivity. So something is happening at this interface on top of something happening at the iron cluster, triggered by something way over here. Is that unusual in biology? No. I mean, think about the ribosome. EFTU, messenger RNA, tRNA, way over here. That's what nature does. She controls all this by conformational gating, which is really hard to study. Yeah.
So
Yeah.
I think when you're 70, you can be asked to try to answer a more philosophical question, which sort of follows from what Don
Yeah.
has asked you, but your last bit of what you just said sort of defeats the
Yeah.
question. And I was going to ask you, why so long? Why so much distance? I mean,
Okay,
hang on.
Yeah.
I mean, you don't need the distance because other
Yeah.
RNRs
So
have it.
it's called regulation, and I don't really understand that, but with the class 1A RNR, you can do 1 million turnovers, and the enzyme does not kill itself. That is amazing. That's because the landscape is uphill. You don't, so it protects itself. You have these really hot oxidants, right? How does it prevent itself? It does 1 million turnovers, it doesn't die. With a B12 enzyme, it dies in 1 in 1,000 turnovers. So there's something, B12, I mean, if you look at all the B12 literature, where's Squire, he left, but if you look at the B12 literature, basically you have chaperone proteins that remove the cobalt 2 states and the cobalt 3 states to
So you think the long wire is protective?
I think it's regulatory. I don't know how, but it's conserved. I mean, even though we do different initiators, like they see with the 1C, right, with no tire, so the details have to be different, but the pathway, I think, is going to be pretty similar, and is regulated by substrates and effectors. Yeah.
So, with the structural similarity of the catalytic portion
Yeah.
suggests to me that these different ways to generate the thioradical were sort of evolutionary overlays
Yeah.
on something that existed initially, and you can think of reasons why that would be if the first enzyme was oxygen sensitive and it started to die. So maybe the long distance or the weird B12 binding site, they're all just reflections of what happened to get those systems.
Well, even the B12 binding site, it's still strikingly similar to the class 1A.
Right.
And it's this movement, this is what you guys need to think about, it's this movement, you can see these things move that I guarantee is a pathway. And you know what it also looks like? Think about this. I can't do this anymore. But think about this. MMO, if you look at MMO, you have a dye, everybody compares X to Q, right? There's a B. Okay, B is key to MMO reduction in chemistry. B looks a lot like that part of alpha. You need to go back and look at that. I'm convinced that there's something there in terms of the communication across that interface, and somebody needs to go back and look at that. Yeah. All right, let's go.
All right.
Oh.
Everybody, let's